Hi, my name is Pia Addison from Stellenbosch University, and this video is about sorting and identification of fruit flies. So this webinar will deal with how to identify and sort fruit flies from various monitoring activities. It is specifically focused on South Africa and Mozambique and forms part of the Fruit Fly Free project funded by the STDF. Trained technicians in charge of fruit fly monitoring will empty trap contents in the field and transfer them to a suitable container. Remember to wear rubber gloves when handling lures and toxins such as Vapona strips. This is important if you are using different lures. You do not want to cross-contaminate the traps. Also use gloves to handle the traps. If you get any of the lure on the outside of the trap, the flies may not go in. Vials must always be properly labeled with the information as indicated above. You should always include the host if your samples were reared from a particular plant. It is best to place the label inside the vial as well, as the outside often fall off or smudge. Use a pencil or pen with insoluble ink for this purpose, preferably on high-quality Teflon paper. You will need a coding system to give your trap a unique reference number. Most institutions have such a system in place. Note any observations in the field about your trap, such as if it is missing, damaged, or has been moved. Any information that is relevant to explain the trapping data is important. Each trap sample should be kept separate. If you caught nothing, please note down 0. Do not leave the field blank. Keep the samples cool and process them as soon as possible. If you cannot process a sample soon after collection, store them in 75% ethanol. When sorting samples, first separate fruit flies from other insects. Soft tweezers and a fine paintbrush are best for sorting flies. Non-target specimens can be kept depending on if they are useful data to have. Otherwise, they can be discarded. We recommend keeping all insects in a first phase of monitoring for quality control purposes. Sorted fruit fly samples should be identified to genus and species. You can use the fruit fly identification key developed by Virgilio et al., which is freely available on the web and indicated on the top right corner. The first step in using the key is to use subsets. You can choose a subset by clicking on the diamond shown in the red circle above. Here you can choose to see only flies for a specific region or using only easy characters, for example. If you select a region and this does not give you an unambiguous ID, select all adult species or do not click any subset. You are now ready to start the key. Select a genera if you know what it is, or click genus unknown if you don't. Please view the specific webinars on fruit fly identifications for more information to help you. A tip is to use the Magic Wand tool or Best tool shown in the circle. This will give you features that are best to use to get an identification quickly. When you have finished identifying all individuals in a sample, you can return them all to the same vial if they are all in one species, or if they are different species, only if there are not more than 100 individuals in the sample. Flies are stored individually if they cannot be identified and need to be sent to a specialist. For preservation of flies and shipping protocols, please see the specific webinar which deals with this topic. It is vitally important to keep good records of your identifications. A specific format is recommended, which includes details of traps, the monitoring data, voucher samples, and monitor details as indicated in the yellow box. The datasheet for trapping details will record the unique trap ID and the abundance of the different species found in that trap. It is also good to record when the trap was baited, serviced, and who serviced the trap, as well as who identified the samples. Anything that cannot be identified or that is not found in Southern Africa must be forwarded to one of the 3 regional fruit fly taxonomy centers indicated above. Samples should be sent via registered mail or courier. See our specific webinar for preservation of fruit flies for further information on shipping fruit flies. The regional centers may use the LUCID key, other reference works, or molecular techniques and provide feedback to the sender. Anything that cannot be identified by the regional centers will be sent to the International Reference Center in Belgium. Specimens that are thought to be invasive require quick and accurate confirmation. If the first tier identifier can provide high-quality and relevant images to the regional specialist, identifications and verifications can occur very quickly. Images can then be forwarded to the international specialist if required. Relevant images are indicated in the photos shown and include the head, thorax, dorsal and lateral views, legs, dorsal view of the abdomen, and wings. Samples should be labeled with a unique identification code of the sample. If images are not able to confirm the ID, or if the status of an invasive species is confirmed, the material should be forwarded according to the second and third tier procedures from the previous slides. Sending images can also be considered if only a limited number of specimens cannot be identified. It is also suggested that vial batches be sent to the regional centers to check the quality of sorting and identification process. During this process, the full data sheets are sent to the regional centers so that they can mark the specimens that they would like to receive for checking. This is a way to determine if there are any misidentifications or issues in the taxonomic process. The number and regularity of samples sent to regional centers depends on the diversity of species caught, and amount of sample traps can be determined for each case separately. In case no adult emerges, the pupae can be stored in 75% ethanol. Remember to always provide the unique identifier, which is linked to the relevant data sheet. Adult samples can be processed the way as described earlier for trapped samples. Larvae can be sent to the regional specialist in Stellenbosch, or the multi-entry key for fruit fly larvae can be used. It should be noted that this key requires dissection of mouthparts from the larvae, as well as measurements. Please view the separate webinar on this topic for more information. Thank you for your attention.